Hello and welcome. My name is Victoria Deary, and today I'll be sharing with you the project I've been working on of quantifying the copy number of variation of the mutated ACE1 gene involved in pesticide resistance in the crustacean Hyalella azteca, pictured in the top right. Hyalella azteca are part of the base of the food web in the San Francisco Bay Delta, California, as well as widely used for sediment toxicity testing due to their sensitivity to a wide range of contaminants. However, populations of Hyalella azteca have become resistant due to pressure from increased pesticide loading entering their waterways after storm events. And normally, pictured in figure 1 with a red X on it, acetylcholine esterase is degrading acetylcholine, pictured with the blue triangles, stopping the nerve impulse as necessary. However, with the pesticide inhibiting acetylcholine esterase, acetylcholine stays bound and the organism can die through neurotoxicity of constant neuronal firing. However, some Hyalella azteca were found to be resistant, and in some part, this has been shown to be because of a point mutation in the ACE1 gene involved in pesticide resistance. So as you can see in figure 2, this is a hypothetical of a mutation indicated by the star. A mutation can occur in DNA, and that gets transcribed into a mutation into the mRNA. And since it is in the first nucleotide of the codon of the mRNA, the amino acid produced is actually changed from a glycine to serine, which is a nonpolar to a polar amino acid substitution, which changes the folding, shape, and functionality of the protein, conferring resistance for organisms that have this mutation. However, the assays done on mutated and non-mutated populations of Hyalella azteca have indicated that mutated ACE1 gene provides less functionality than the non-mutated wild-type gene, leading to believe that the mutation of the ACE1 gene has less functionality than the wild-type. So we believe there is more at play than a simple mutation for the resistance of these organisms. So we hypothesize that copy number variation is a crucial role in the resistance of these organisms. So in figure 3, in the top there's the reference gene, and below that is an example of the gene copy number variation, genes indicated with a box. And Hyalella azteca is a diploid organism, which means we expect 2 copies of each of its chromosomes, so double of what's pictured in figure 3. In figure 4, Kaylee Major took clones of representative individuals from different populations and sequenced them to determine alleles. And as these are diploid organisms, we would expect 2 alleles, all the bars reaching to number 2. However, you can see here that there is definitely copy number variation. The blue bars indicate the mutated alleles of the glycine to serine, and the orange bars indicate the non-mutated wild-type allele for ACE1. And now we would like to quantify the number of copies and relate that to how resistant the organisms are, as well as how functional their ACE1 gene is. So in order to quantify how many copies of this ACE1 gene the organisms have, we first made sure to find genes that were in single copy so that we could adequately compare the two. So figure 5 shows the sequence of what we did to arrive at those single copy reference genes. So the first step was to look at the Bosco's list of conserved arthropod genes, BUSCO stands for Benchmarking of Universal Single Copy Orthologs. From that list, I selected 10 genes, and from there I looked into literature to see if others have used those genes in copy number variation experiments. And I also looked into the function of those genes and wanted to ensure they provided essential function, which further indicates that they are likely in single copy as well as present in my organism. And to further indicate that, I then I took the genes from the BUSCO's list and I found their homologous genes within the Hyalella genome. From there, I looked at the genome for highly conserved regions of these genes so that I could then design primers to those regions so that these experiments would be applicable across multiple different populations of the Hyalella azteca. From there, I did preliminary qPCR experiments to determine the product length, as well as the genome itself indicated the product length that we should be expecting. And the qPCR indicated the melting temperature of the genes, which was then used to optimize and set up the qPCR experiments for maximum consistency for each of the reference genes. And the qPCR also told us that there is one product made for the genes being used, which is important for us to then relate back the copy number of the ACE1 gene to these highly specific, highly conserved, single copy reference genes. So in figure 6, you can see 2 of the genes that were selected as reference genes. Originally there was 4, but after further experimentation as well as an RNA extraction and sequencing, we found that the RAN binding domain, RAN-BD, and the RNA polymerase subunit 10, RPB10, are the single copy reference genes that will work best for our experiments. So in conclusion, the RAN binding domain and RPB10 are useful, highly specific, single copy reference genes that have been shown to provide us with consistent efficiency and single product across the different populations of Hyalella azteca used in this study. Using those genes as the single copy reference, I've repeated the qPCR experiment with primers made for the ACE1 gene. Comparing the two helps to identify and quantify the number of ACE1 gene copies in each individual Hyalella azteca. Figure 7 compares mutation and copy number variation. So this shows the haploid number, which again, we would expect to be 1 if the organism's gene was not duplicated. However, non-mutated ACE1 gene having Hyalella azteca indicated in blue have above the 1 mark for their haploid copy of their ACE1 gene, and mutated ACE1 gene having Hyalella azteca indicated in red have even higher numbers of their ACE1 gene. This seems to indicate that both mutation and copy number variation lend in Hyalella azteca to become extremely resistant to the organophosphate pesticides. We will hopefully be able to correlate, or at least determine, how the mutation, copy number variation, and functionality of the ACE1 gene are related to each other. And the latter part will be the next step of the project using an assay. The last figure indicates that the mutated duplicated gene and the wild type copy allows the organisms to both be resilient against pesticides as well have functionality of their ACE1 gene. The point of this project is to hopefully urge others to decrease their pesticide use, as well as to inform regulations for toxicity standards,
to consider evolutionary biology in sediment toxicity testing, since these organisms are used to set the baseline of health and safety standards for people swimming in these lakes that Hyalella are inhabiting, or even eating fish out of these lakes. It's important for us to then understand what is affecting the organisms that we are using to set these standards so that we can make better regulations and lay people can make better decisions of where they swim and what they eat. And I would like to thank my PI,
Dr. Helen Poynton, as well as lab mates and Nadira and others who have provided experimentation and funding, such as IMSD, NIH, and the California Department of Fish and Wildlife. Thank you for your time.